Hello, and welcome to the program. My name is Sam Ankerson. I'm the Executive Director of the Bailey Matthews National Shell Museum and Aquarium, joined by Dr. José Leal, who you can see there on your screen, our Science Director and Curator, and Associate Director of Education, Jorden Falker, who's right there, and we'll see more of her later. And thanks for joining this program wherever you may be. We're glad to be able to share this subject, some of the, I guess, call it behind the scenes work that happens here at the museum, and important work that happens in addition to uh, exhibiting mollusks and shells for visitors and creating educational programs about them. It's important for this museum to participate in work that advances the general body of knowledge about mollusks and shells, their habitats, how they're doing, and and a whole range of things. So what you'll hear and see from Jose and Jordan with this program are some examples of that. In some cases, it's research projects with through national and international partnerships. In some cases, it's getting an assessment of local mollusk species here on Sanibel with volunteers and enthusiasts and beachgoers. So we hope you'll enjoy it. This is the first program of our summer fall Zoom lecture series. And I'll quickly mention the other three. On August 21st, a program called Seasonal Florida Seas by photographer and photo naturalist Gabe Jensen, an amazing photographer who happens to be one of two featured in a new exhibit at the museum right now, underwater photographs of nudibranchs. But they're terrific photographs, and also a kind of a narrative about how, as the seasons change, the dynamics that happen in the oceans and how they affect marine life. On September 18th, our friend Steve Geiger from the Florida Fish and Wildlife Commission will be presenting on the latest in sort of the connection between oyster populations and in the Everglades, and how that relates to restoration projects in the Everglades. And then on October 16th, John Fallon, who's Director of Sustainability at the Audubon Nature Institute in New Orleans, will be presenting on the broad topic of seafood sustainability in the Gulf region. So these programs are all free, and you can sign up for them just the way you did for this one through the museum's website. And we hope you'll join for some of those. And also on that on the website as well is an archive of all of our past recorded Zoom programs, and there are well over 25 now, dating back several years, which is a great resource, and those are always accessible. This program is delivered via the Zoom webinar format. So it's not like the Zoom meeting where obviously, we're not all sort of here at the same time in a sense. But with questions, we'll answer questions after the presentation, but please feel free to ask your question at any time. And the way you can do that is by using the chat function. And to access that, you run your cursor along the bottom of your screen, and you'll see the chat button there, and you can just type in your question, and we'll gather all those and answer them afterwards. So with that, we'll turn it over to Dr. José Leal, who will start who will sort of do the first lap here. Yeah, Jose is the science director and curator, and has been with the museum since 1996. He is one of the preeminent malacologists in the world, and a fantastic curator, and great leader of this museum. And then following up, José will be Jordan, our Associate Director of Education for 2 years now, I believe, or going on 2 years, and has been with the museum for over 5 years. And among many hats she wears here, she leads our programs in community science, which you will hear a little bit more about. Okay, thank you. Take it away, José. And you're on, you want to unmute yourself there, José.
All right. Yes, you can hear me now. I want to thank Sam for his kind words, and thanks to everyone in the audience for joining us today. What we want to do today is just give you a brief overview of some of the research, some of the traditional research we've been doing at the museum for quite a while now, using collection materials, but also introduce some of the more recent field of aquarium-based research that is taking place here, and that hopefully will go on for quite a while. A lot of the themes I'll be showing you today are locally themed, meaning we're working with local species in the Gulf and Florida marine species, but some things go a little beyond that, as you will see during my presentation. The first topic I want to show you is a paper going back to 2023, work we've done based on a very cool time-lapse video taken by our senior aquarist, Holly Hulse. And she took a video taking frames over a period of 113 minutes, basically almost 2 hours. And she compressed that. We doing using techniques, in the actually the iPhone, compressed 113 minutes into 27 seconds of this awesome video of a female crown conch in our aquarium laying egg capsules. And you have a chance to see now if you haven't seen that yet. The paper was published as a little two-pager published in the Bulletin of Marine Science out of my alma mater at the University of Miami. So let's take a peek and see what happens here. What happens here, you're looking at a female crown conch clinging on to the inside of one of our aquariums. And as you see in this long, expansive time of 113 minutes condensed into 27, you see the crown conch laying one egg capsule. You can see that there are egg capsules already laid here and glued, quote unquote, to that plexiglass wall of the aquarium. And you'll have a chance to see her laying yet another egg capsule. And just after that, moving one packet of eggs into a gland that sits on the bottom of the foot here called the capsule gland. It's called the capsule gland because it's a gland that makes egg capsules. And after that, the video stops, but naturally this egg capsule laying would have continued in the tank. So without further ado, let's take a look. So what you're seeing here again is a very compressed view of what happened in almost 2 hours. Not much is going on up to a point very close to the end of the video. Right now, well, I'm going back here, but you will see at some point, and I will call your attention at that moment, for the snail gluing one more egg capsule around here. Do you see that? This is the new egg capsule that was just laid up. And now a packet of eggs came out, went inside the capsule gland, and the snail is making a new package of capsule around that to be again laid down onto the plexi and so on. So the cycle continues. Another line of research we're following here relates to the non-indigenous, the introduced species that many of you now know about, the Thrush Cowrie. The Thrush Cowrie was found in the early 2020s in the Southern Caribbean, I guess in the island of Aruba. In 2022, I wrote this little piece in my blog in the museum website. An invasive mollusk makes headway in the Caribbean. Where next? And my hint here in this short article was that the species would show up in Florida sooner than later. And lo and behold, um, in early 2023, in a little paper led by Anton Olenik, a colleague at Florida Atlantic University in Boca, across Florida, we joined forces and published a little note in the Nautilus relating the first finding of the thrush cowrie in the USA, finding of the species in Palm Beach County uh, back in, I guess the first finding took place sometime in late 2022. And since then, we've been accumulating a lot of data, and I've been putting out little write-ups in my blog and elsewhere in social media about that. This is a photo of one specimen, a live specimen, live collected by Ken Pietz in one of the islands near Key West in the Florida Keys in Florida. And that was collected back in August 2023. In 2024, the species was found in the Florida Panhandle in Panama City Beach. And that's really strange because there is a big gap between, the closest spot where the species was found to the Florida Panhandle is Key West. There are no findings of the species between Key West and Panama City. However, there are many reports of the species in the Florida Panhandle extending all the way to Mobile, Alabama. I've been putting announcements, requests out there for anyone that finds the species anywhere off the west coast of Florida and Gulf of Mexico to let us know so we can see what's going on. And ideally, we'll be trying to get genetic profiling of those specimens to see what is more related to what in this whole landscape of the introduction of the species. The species has been found in very large numbers. A couple of weeks ago, I published this little blog entry about the species being found in in huge numbers by a colleague, Dr. Darren Goldman, who is an ophthalmologist out of the east coast of Florida. In a single snorkeling excursion, he found around 150 dead shells of the species. We are pursuing this with colleague Anton Olenik, Janessa Fletcher from there, who works at the Florida Wildlife and Fish and Wildlife Research Institute out of St. Pete, and Alex Fogg, who works for the county of Okaloosa in the Panhandle. Alex also has found huge numbers of the species off Okaloosa Island Pier and neighboring areas in the Panhandle. Here we go. A couple of more pictures of the live animal to give you, a good image, search image for those of you who want to look for the species on the west coast of Florida. Another cool project that ended up in a nice publication was to determine the true identity of the Florida conch. Back in April 2022, an author published an account showing that the type of Conus anabathrum, which is the name we have applied to the Florida conch for many years, that the type, which is the specimen that's used in the original description, that type would not be a shell of our species, but however, a shell of a species that lives in the Eastern Pacific Ocean, Conchs calaris. One of the main problems with this type is that it carries the name Conchs anabathrum, but it doesn't have a locality associated with it. It's in a museum, and all it has is the name and the shell shape. And this author was claiming that that type is not the species we have. So that name, Conchs anabathrum, could not be applied to the Florida species. So we took the project head on with colleagues, Dr. Greg Herbert from the University of South Florida, Bill Fenton, um, Aaron Avery, who is a student of Greg Herbert, and Federico Marquez, who is a colleague who does very sophisticated geometrical analysis out of Argentina. Actually, he lives in Puerto Madryn, which is in Patagonia, and works in one of the really well-known research institutes in that area. So we did the work, did some of the geometrical morphometrics on the two different species, and tried to place that lost type into either one of the species. And naturally, the type fell into what we call the morphospace of the Florida species, showing that indeed the name Conchs anabathrum should be the name applied to the populations of conch from Florida. A little paper again in the Bulletin of Marine Science, and this one put out by yours truly, again, Carly, Claire D'Agostino, who is our ambassador aquarist, and our contract veterinarian for the aquariums, Susan Fogelson. We looked into a problem that happened when the aquarists purchased 4 horse conchs for our living exhibits. We noticed that out of the 4 horse conchs in that shipment that came from the Florida Panhandle, 3 of them were females that had a little male organ that we call a pseudopenis. And that's an indication in gastropods of a condition called Imposex. And Imposex is a quote unquote disease induced into marine snails by antifouling paints. Those paints are mostly banned, but who knows? Sometimes people use them illegally, if you will. We don't know if anyone is doing that in that area. Those 4 horse conchs came from an area called Alligator Harbor in Franklin County, the Florida Panhandle again. We reported that in the Bulletin of Marine Science, as I said, providing illustrations of the females with the little incipient male organ, and actually conducted what we call histological sections. I mean, taking very thin sections that are stained, photographed under the microscope to show that those little male organs actually had a duct that we call the vas deferens. And again, in 2 different kinds of orientations for those sections. And now the word is out, and we are asking that everyone who knows or everyone who found horse conchs with that problem to let us know. So we'll try to pursue more research on that. What's the importance of that? The problem with Imposex in many marine snails that it hampers the ability of females from reproducing. The females are no longer viable. They cannot have babies because their systems become all compromised by that deficiency. This is really becomes a real problem with horse conchs because as Dr. Greg Herbert and his collaborators in 22 found, the lifespan of horse conchs is really short. They are known to live no longer than 16 years of age. 16 years would be the lifespan of the world record horse conch shell that we have here at the museum. In addition to that, horse conchs have the females they show what we call delayed maturation. So they can only have babies after about almost half of their lifespan has passed. So they will have babies later in life. So they have a very short window for reproduction. You know, having children between 6 to 7 years um, after they, you know, after 6 or 7 years of age, and with large females spawning only 3 to 5 times in their lifetimes. So that restricts the reproductive output of the species, presenting a problem for for the species for the conservation of the species, and that conservation problem is emphasized by Imposex. So the call is out for more research on that, and from other areas, you know, checking out to see horse conch living in areas where antifouling paint is used if they are coming out with a problem. Yet another research project that actually we had a long meeting today about that earlier on is a project where we are looking at a very interesting question, is whether the abnormal right-handed lightning whelks, as you all know, normal lightning whelks are left-handed, but there are quite a few right-handed lightning whelks over the entire range of distribution of the species here in Florida and elsewhere. Instead of having the aperture on the left-hand side, they have the aperture on the right-hand side, and you can see an example here. So what we're doing is we're photographing them using exactly the same orientation for all photographs, and then doing, again, using techniques of geometric morphometrics to see if after we reverse those pictures, like I did now, I went back and again, I'm going to do it again, reverse that, and then we apply the techniques and do the statistical comparisons to see if the right-handed whelks, when swapped to a left-handed condition in a photograph, if they have cells that can be compared, and whether there are differences between the right-handed and the left-handed when the right-handed are reversed. And we're trying to find also whether there are symmetry differences in the shape of the animal, in the distribution of organs and all that. So that's a very cool project. And right now, we've taken pictures of about 19 right-handed lightning whelks. And the project continues. I'll let you know more as we go. This has not been published yet. It's a study done by a student at the Hawaii Pacific University in Honolulu, Hawaii, Allison Nims. And I joined forces with my Hawaiian colleagues in looking at the identity of some larval gastropods that are found in surface slicks. Surface slicks are parts of the ocean, especially in shallow water, where what we call internal waves, which are waves that you cannot see, but they're underwater, big masses of water that kind of collide and diverge. And when that happens, the surface of the water changes its density, and you have a strong accumulation, a very dense accumulation of small marine animals, including mollusk and larvae. So we looked at a number of species of those larvae, and the group in Hawaii did the DNA sequencing and put names to those larvae, meaning they made the connection between the DNA of those larvae and the DNA of grown-up gastropods deposited in a huge database called GenBank. And they found some names, and I helped with the morphological identification of the larvae to confirm that. And that's the poster presented by Alison Nims, who is a student of my colleague Brandon Hall and a professor at the Hawaii Pacific University, and Jonathan Whitney, who is a NOAA biologist also in Hawaii. So let me show pictures of those larvae. This is supposedly the larva of a cone species, a drupe that is a murex species, and a moon snail called the Gualtieri's moon snail. Those are pictures of the larvae, and I'll show you pictures of the most closest related species to this larvae. This is actually exactly the same species as that. And can you look at the difference between the larval shell of this little murex, muricid, a prickly spotted drupe. That's the larva. That's the adult shell. It's pretty cool, isn't it? So when comparing DNA, you can match larval shell or larval organism to the adult. The last thing I want to show you is just so many of you know about our shell guide. And I think that the shell guide, in my humble opinion, is probably the best research product produced by the museum. And I hope it will last many, many years. It's a resource used, as I wrote there, by shell enthusiasts, fisheries biologists, ecologists, citizen scientists, conservationists, and all my museum colleagues. It's important that we know what's here, exactly their names and what they do for a living. I just want to quote this Baba Dium, who was a conservationist that was with the International Union for Conservation of Nature, said, in the end, we'll conserve only what we love, and we will love only what we understand. So I think the guide is a great resource for understanding, the mollusks that live around here. Next, I want to invite our esteemed Associate Director for Education with the National Shell Museum, Jorden Falker.
Hi, I'm Jordan. Now, we're going to take a moment here to share Jordan's slides. So let me just pull these up. Sorry.
How did we go so far ahead?
Okay.
There we go. Okay. I'm gonna say, do you care if I move the laptop more? Okay. All right. Move it all the way over. Okay. Hello, everybody. So my name is Jordan. I'm the Associate Director of Education here at the museum. Most of my job is running a lot of our educational programming, but part of that is overseeing our community science projects. A community science project is essentially a project where the public is involved in data collection or fieldwork or analysis, various things. And the museum has two that are currently going on. We have live mollusk counts and we have a snail search of Lee County. So, all right, live mollusk count. I'll start with that one. Live mollusk count first started in, and correct me if I'm wrong if you're in the Shell Club and you know that this is not the right date, in 1962 by the Sanibel Captiva Shell Club. And members of the club, essentially, they went and they searched the beach for living mollusks. And then, to my knowledge, it was not conducted again until January 2022. So Joyce Mathis, she was a longtime volunteer here at the museum and a member of the Shell Club. She kind of led the charge in 2022 for this count, and they did the same thing. They recorded their findings on data sheets, and then later in 2022, we all know what happened there. After Hurricane Ian, we started to partner with the Shell Club for an island-wide live mollusk count every January. And you might be wondering why January, and that is because the lowest tides of the year are typically in January. We have some very low tides. So in live mollusk counts, people walk their assigned stretch of beach in the designated day and time. We figure out ahead of time when the best tide is, best day and time. They go out, they look for live mollusks, and they record their findings on data sheets. They take photos of anything unusual, super interesting, whatever they want. And these walks are always conducted at very low tides. We never do them at high tide. And then beginning last year, the museum began leading live mollusk counts every other month to gather more data. These ones are not island-wide. This is only focusing on Lighthouse Beach. Oh, why are we doing this? It's fun. I mean, yeah, it's fun to go out and look on the beach for live mollusks, but why are we doing it? The January counts, we want to see what species are being found year after year, what ones we maybe go a while without seeing. We want to see how the east end of the island compares to the west end, then compares to the bay. And then the year-long counts that started last year, we want to see how the same beach is different throughout the year. If weather plays any role, if temperature plays a role, any impacts from red tide or other hurricanes, things like that. And ideally, we want to gather a large data set. So this is just the beginning. All right. This is just to show you the Lighthouse Beach stretch that we cover for the year-long walks. We start at the Lighthouse Fishing Pier, and we walk all the way down to the Colony Inn beach access. All right, so here is some of the data that's been collected so far. And I'll make a note now that none of these charts account for observations described as hundreds, thousands, infinite. If you've been on a live mollusk count with the Shell Club or with the museum, sometimes you go out there and you find so many mollusks of a certain species that you just can't count them all, and you just make a note that says hundreds or 100 plus. This data does not account for that. It's pretty hard to put on a graph. So the bar chart on the left, data in 2023 is not pictured for either of these, just because I did not have access to the different sections of the island's data. I only had the final numbers for the entire island. So 2024 is really when I started to be a part of these of this live mollusk count project. For the east end, we define that as the Lighthouse Fishing Pier to the Tarpon Beach access. In 2024, there were 886 mollusks observed, and in 2025, only 250. The west end of the island, we defined as Tarpon Beach access all the way to the end of Blind Pass. And in 2024, there were 338 observed and 426 in 2025. And then the bay, we define as the Lighthouse Fishing Pier through Bailey's Beach Park and the little beach at the end of Dixie Beach Road. So, and with that, 159 observed in 2024 and 51 observed in 2025. So you might be wondering, especially when you look at the difference between 24 to 25 on the east end, it's a pretty large difference, much smaller number this year. That doesn't really tell us anything right now. And the line chart on the right, so one thing I'll point out is that the March count last year had to be canceled because of severe storms. So we ended up going out in April. But this year we went out in March. So what was I saying? Oh yeah. So January of 2024, only 26 mollusks observed for Lighthouse Beach. That the one group did not walk the section. That was a rainy morning. There was lightning for the first part of it. That can account for a small number. We don't know. Maybe they would have found more. We're not really sure. And then in April, it jumps up to 121. In May, goes down, obviously. July, September, November. So you can follow the blue line there. That's 2024. And then in 2025, obviously you see there's a chunk of it missing. This year isn't done yet. But this year in January, only 37 live mollusks were observed at Lighthouse Beach. And that's where everybody walked and the weather was beautiful. It's kind of cold, but it was beautiful. So you can see in March, it jumps up to 186. And then we take a pretty dramatic drop in May. And then July's live mollusk count was just on Monday evening. And we observed 66 living mollusks. So as you can see here, so more living mollusks were observed at Lighthouse Beach in 2024 so far than this year. But this year's not over. And like I mentioned, this is kind of the beginning of a much larger data set. So it's definitely too early to be drawing conclusions on what this means. So here I just want to talk about, so some fun things. These are photos that have been sent in for live mollusk counts. These are a great opportunity to see really neat things on the beach. So the top left photo there, that is a white baby's ear. The bottom left, obviously, is a true tulip. And then on the right, that is a living horse conch. And it has a living line, oh, I'm sorry, is an eastern beaded chitin on the shell. The troubles with community science, there's a lot of room for human error. The groups, everybody walks at their own pace. They have different methods of looking for live mollusks. If the weather isn't cooperating, they might delay their start or end early. Rainy weather can lead to a group walking faster. They want to finish their search in their section of beach and they want to get off the beach, so they might miss things. So all things to consider when looking at this data. All right, the other community science project we have going on, this is Snail Search of Lee County. Snail Search is where people take photos of living land snails or evidence of land snails, which is essentially the shell. And that's in the order Stylomatophora. So that covers most of our land and tree snails and slugs that you find here. But there are certain slugs that are excluded. Like we have, I think it's called a leatherleaf slug. They're not in order Stylomatophora. They don't count. And periwinkles are not part of this either. So after people take these photos, they upload them to iNaturalist. So we use iNaturalist to keep records of all of the observations. They're automatically uploaded to the project page. And this project started in 2022 on November 7th, which is actually International Land Snail Day. So this community science project, as far as ability to participate, is much more relaxed. You don't have to go out at a certain day or time, and you just go out whenever you want and find snails or attempts to find snails and record it. So Snail Search, we decided we wanted to do this. Well, first we saw a very great community science project done in LA. She actually, Dr. Jan Vendetti, she did a online lecture for us a couple years ago about the community science project with land snails out in LA. And that got us thinking, what do we know about land snails in our area? And the answer is not a lot. There are not many studies done in this area. One of the only ones that we know of was done in Ding Darling in the early 2000s by the late Harry Lee. And it has no images. So with iNaturalist, it's a very image-based project. And so the other reason we decided to do this, we wanted to get people interested in snails. A lot of people think snails, land snails in particular, are icky, or they think of them as garden pests, but they're pretty important to the ecosystem, and we'd like people to, kind of appreciate them for what they can do. So here is a map of the county. So the highlighted in yellow portion, obviously you can see that is the boundaries of Lee County. By this point in the project, we've reached almost all the corners, I think. We've got a pretty wide area going for us. as of June 30th of this year, we have 247 observations total, and we've had 111 different participants in the project. So over 100 people have photographed a land snail or evidence of it in Lee County and have uploaded it to iNaturalist, which is great. So these are the top 3 observed species. The large photo on your left is the southern flat coil. We've had 82 observations of this species to date, and this is a native species of land snail. The top right, that is the Cuban brown snail. We've had 80 observations, so pretty close to the flat coil, but this is considered a non-native species. And the bottom right there, that is the ghost bulimulus, and that is 39 observations. So kind of a dramatic drop, 80 to 39. And that's also a non-native species. So you may be wondering, this seems kind of fun. How can I participate in community science with the museum? Well, I'll tell you. So we have our volunteer page on the website. That's what this photo is. If you scroll down, we have a new position for community science volunteers. You don't already have to be involved with the museum in any way. You do not have to be an expert on snails or aquatic mollusks. You just have to want to get out there and help us get this data. So you can do it that way. Or with Snail Search of Lee County, you can just go out, make an iNaturalist account. It is free to make an account. You can go out, take pictures of snails, upload them to the project, and there you go. You've participated. It's as easy as that. With live mollusk counts, that's what, if you're not interested in volunteering year-round, maybe you want to try one out, see if you like it. We do host them every other month, and the information for those are also on our website under the Learn and Experience tab. You'll find it under Community Science. So that is the end of my slides, I guess. So here's Sam.
Thank you, Jorden, and thank everybody. And José, let's come back on camera so we can see your smiling face. And we'd love to take any questions. And I'll go to the check on José. We'll go to the chat room here, and here, Jorden, here.
All right.
And I know we had at least a question from Western Australia, which is amazing. Okay. So, yes, this is Taryn Ryan, who says, "Good day from Western Australia." Thank you, Taryn. That's fantastic. So this relates to the Imposex item, José. Question is the anti-fouling paint being spilled wet or coming off hulls / leaching into the water after it's set on the ship?
Good day, Taryn. We really don't know. I mean, this is a very remote area of Florida for us here. We're probably I would say about 500 miles from here. And it is an area that has, a series of shrimp fishing fleets, small fleets of shrimp trawlers. We have no idea what's going on. It could be that it could be either of the conditions you're mentioning. It could be spilling wet, during the process of, painting the holes out of the water, or it could be leaching into the water after it's set on the structure of the ships. We really don't know much about that. We just recorded that first, biological effect that we suppose it's coming from what we call the organotins or organotins of used in anti-fouling paint. I don't know if I answered. I mean, the answer is that we don't know.
Uninformed, we don't know.
Yes.
Yeah, thank you. Okay, 2 questions here from Minori. One was, apologies if I didn't catch this. What was the result of comparing left-handed and right-handed whelks?
Well, that's a project that just, we started at some point late last year, and one of our colleagues had problems and is just coming back this week. So it's a project that has, I would say, it's about 1/3 in progress. So we don't have any results yet. We'll let everyone know. It's a brand new concept that we're beginning to develop.
Right.
Thanks for your
And
question.
here's another question also about maybe not a brand new concept, but a newly understood development. Are there any efforts to manage invasive species of the thrush cowries?
Um, well, first of all, we suppose that the thrush cowrie could be classified as an invasive species. However, there are very rigid classifications that go on when we have a situation like that. An invasive species we have to prove that that species is causing a harmful effect either onto similar species that are native or to the environment. And we don't have evidence of that. However, it's pretty, the hint is there that we have so many that something could be wrong. Now, if we find that there is something detrimental going on, I think something that could be done is something similar to what's done with the lionfish. So it's basically ask divers and everyone who can to go and pick them up and remove them out of the water. However, the number is just staggering. I've heard reports late last week that people are going in the water and collecting, like anywhere between 1,000 and 2,000 of them in one
Wow.
dive.
Oh my gosh. Wow.
So that's something that doesn't happen with the local species of cowries. The local species of cowries are similar size of the Atlantic yellow cowrie, a species in the same genus, Naria acicularis. And I never heard of anyone going, in my, whatever, 50 plus years of diving, and never heard of anyone going in the water and collecting 1,000 yellow cowries. So we may have a problem there. The
Wow.
environment, yeah, but we really don't know what to do about that at this point.
All right. Thank you. Just one, I noticed a couple of people are raising their hand. We won't be able to kind of quote call on you in this Zoom format. So if you have a question, please click on the chat button there and type in your question, and we'll get your question that way. Claire has a comment about the live mollusk counts, suggesting recording wind speed and direction for a few days before the counts.
Good idea. We do record the wind direction and speed the day of the count already, but not beforehand. So that's a good suggestion.
Good thought. Next question. What are the possible, well, okay, so we get this a lot related to the mollusk counts. What are the possible causes for the decline in the number of living shells?
Well, one thing that could contribute to it is red tide. We know the impacts of red tide on living mollusks. As far as other causes, that's partially what we're hoping to accomplish with this. If it's severe weather, if it's increased tourism on the beach leading to more live collecting, we don't know.
Yeah, and I might say, Jose,
Yeah.
sort of adjust or correct if this is misstating, but I think we're with the mollusk counts, we're excited to sort of be more methodical about collecting data, but I think to really draw conclusions, that you could kind of hang your hat on, there would just need to be a lot more data over a long period of time. So we're my point of view, we're just sort of in the first steps of this, of taking this great idea initiated by the Shell Club and just looking to try to do it year round. But we're just sort of gathering numbers and sort of sharing them and looking at them, and I don't know. Would you agree, Jose? It's only with
Yeah.
time and a lot more can sort of draw conclusions with any more confidence.
Yeah, I think you're right. I mean, the effort is great, and we get a lot of good data. We have a lot of things that we wouldn't see. I mean, Jordan, in one of the beach walks, just found a species of sea hare that we never heard of here. This was just early this week, right?
On Monday, yeah.
On Monday. But to draw serious scientific conclusions, you have to have a more structured research plan, and that's not the real goal here. I think is we can do the counts and get some ideas, and maybe use those counts to plan better for future more refined projects. And I think
Right.
that's a great idea. What's going on?
Yeah.
Um,
Yeah. Thank you. Okay. Question from Alan. Can a right-handed whelk mate with a left-handed whelk?
great question. No.
It's been
that,
waiting for that one.
that's why right-handed lightning whelks. Remember, there are like right-handed whelks. There are normal, like someone mentioned the knobbed whelk from the East Coast. We don't have it here. That's right-handed, naturally right-handed. The lightning whelk is naturally left-handed, and the abnormal ones would be right-handed. And if we have a male of a right-handed whelk, he won't be able to properly, match with the female of a normal left-handed female. So yes, no, they can't mate.
Okay. Thank you. Good question. Good answer. Okay. Question from Nancy. How far north on the east coast of Florida have you found Curtis?
That's the thrush cowrie.
Well, the
Thrush Cowrie.
thrush cowrie, yeah, the thrush cowrie. I think the farthest north that I know, and those things change like boom, like every day is a new. It's like someone comes with a sledgehammer on my head, to use a strong expression, but I think it's Port Pierce Inlet. It's where the northernmost occurrence of the thrush cowrie happen. Now, and I forgot to say that, if I have a minute, I forgot to say that in my part of the program, is that this huge gap that we see between Key West and Panama City Beach. And from Panama City Beach, it goes on all the way to Mobile, and it's very, very abundant in that swath of the Panhandle. We think that that was another introduction event that's independent of what happened here on the East Coast. So it probably came with currents from warships, or who knows, from the Caribbean.
Right. Question from Lori. She's for Jordan as relates to the counts. Do we think the Gulf hurricanes have affected the mollusk counts?
I would say it's just too early to tell. I mean, since starting this, we haven't had a really, really bad Gulf hurricane. Well, since the museum got involved after Ian, I should say. So I think it's too early to tell.
Yeah, if I may, in
Absolutely.
relation to hurricanes, we have to keep in mind that hurricanes have happened for a while. It's not something that started happening, since I moved to Florida, which was like 40 plus years ago. It's been going on for centuries, for thousands of years. And the mollusks come back. The ones that are here, the populations have gone through several cycles of hurricanes and non-hurricanes. So I think, as far as their conservation, hurricanes is the least of their problems.
With the most of the problem being probably water pollution, right?
Yep.
Yeah. Okay. Aloha from Maui. Okay, got someone from Hawaii. That's great. Do we know if there's a similar iNaturalist program for land snails in Hawaii?
I mean, I do not know. Maybe Jose knows. He's got a couple colleagues in Hawaii,
Yeah,
but I do not know if there is one.
well, land snails in Hawaii are, their own universe. There is a lot of problems with land snails in Hawaii. Um, and a lot of them have about almost 60% of the original Hawaiian land snails have disappeared for many different reasons. Um, and they do have a very strong program involving biology scientists from the museum, the Bishop Museum, and citizen scientists and enthusiasts that are always out looking for the surviving species. So you may want to check the Bishop Museum website and look for information on their similar programs to the one that Jordan has with us here, and see what they're doing and get information from that.
Thank you. We've got one more question, which is after the beach restoration at Navarre Beach in the Panhandle. Is that how you pronounce it? Navarre? Navarre? Do we know? Beach in the Panhandle. Some people have been finding queen helmets. Do they live that far north?
Yes, they do. But you have to keep in mind that a beach restoration brings, I guess, in your case, in the case of Navarre Beach, that the sand is coming from offshore. They're using big, probably like if it's the same system as been used here. They have big dredges, big barges offshore that pump sand from like 3-4 miles offshore onto the beach. And with that sand, a lot of shells will come too. We have Junonius coming up here, and people waiting for the shells to come out of the big pipes to pick them up. So that may be happening. And we find small queen helmets here on Sanibel Island. Queen helmets are a common species in the Gulf of Mexico in deep water, so I'm not surprised to see that.
Great. Thank you, Jose. Thank you, Jordan. Thank you, everybody, for attending the program and for your great questions. Please consider joining the remaining 3 talks in the series. Next one is August 21st. More information on all of them on our website. And if you are in the area or going to be visiting Sanibel sometime over the summer, please come by the museum. The Great Hall of Shells is looking beautiful with a big chunk of the new exhibits open and the rest of them actually getting installed now. And we'll be ready before we know it. So thank you again and have a great evening or morning or afternoon if you're in Hawaii or Western Australia or wherever you may be. Thank you.
Bye.